Yesterday, in the opening plenary session, both Naz and Carol reminded us that in 2009, the PSOC was founded to explicate and illuminate a grand experiment in convergent science, one in which advocates were embedded in the program at inception. That mission is as urgent today as it was then. Seizing the moment to capitalize on exciting scientific opportunities and the role advocacy plays in PSON cell, tissue, and genome mechanics settings, I hope to convince you of the power of advocacy as a prism for understanding and as a tool for transformation. I will offer reflections on the benefits, deliverables, and key takeaways of science advocacy exchange strategies developed and deployed within the Weaver, Labarge, Odie Flink, and Gartner labs. Among other strategies, the researchers bring their bold ideas and innovative thinking to life for me by inviting me to their lab meetings and conversations. They encourage me to share insights and intersect patient preferences and priorities in their planning and decision-making. The work in the Gartner lab brings together vision and passion for understanding the promises and challenges of thinking differently about the role of innovation in understanding tissue development as a model for therapeutic development. Importantly, the work underscores the challenges that need to be solved in order to build tissues. Irene and Jason in the Weaver lab have been able to show using atomic force microscopy measurements of human breast tissues that the extracellular matrix is stiffer in high density tissues. These data show the story of the patient journey and quality care. These findings suggest that inhibitors of both RANKL and ZNF217 might have benefit in reducing breast cancer risk in women with high breast density. Realizing that it is crucial to detect women at high breast cancer risk as early as possible to enhance treatment outcome, Stefan Hinz from the Labarge lab approaches this issue by studying the mechanical properties of human mammary epithelial cells using novel microfluidic mechano-node-pore sensing methods. So who are we? How do we seize the moment in such preclinical settings? We do so as paladins, as creative champions of science, as cultural brokers and disruptive change makers. Forging credibility in what is at stake in biomedical matters and promoting a comprehensive approach for bidirectional collaboration. I work with transdisciplinary research teams to move it, to shake things up. I actively support team members to think differently about the role of innovation, that it is no longer a solitary, siloed affair. In a changing world, decisions must be evidence-based as well as patient-focused, translatable, and relevant. Conducting research responsibly includes engaging with the public to creatively communicate the research and tell how amazing it is to be a scientist. I'm now leaving you with visual images of our co-creations and co-deliverables, including breast density videos, science art advocacy-driven brochures, conceptual models, and guiding frameworks for establishing bidirectional collaboration, as well as a few specific reflections on our co-presentations and publications. Now, heads turned amongst the scientists at the 2019 CRUC AACR Physical Sciences and Oncology meeting in London, when Carol Boss and I communicated the highlights of Jason Northey's innovative science from the lens of advocacy. This experience was immersive. It put the spectators of our work front and center, allowing them to experience and see their world through our eyes as we describe the relationship between input, output, outcome, and impact. As indicated in these publications, the intersections of science and advocacy is giving rise to new forms of engagement, meaning, and communication that are changing the face of cancer. So what are my key takeaways? We are at an inflection point in the convergence of physical sciences, oncology, art, and advocacy. Collectively, we must work together, transforming insights into action. A creative and innovative advocacy culture has the potential to promote stronger, deeper support and trust in physical science and oncology research.